Good afternoon, and it's good that most of you managed to find your way through the road cones and the don't go this way signs and the other things out there. Either that or it's a cheap way to get parking and then go to the Foo Fighters. Right, well, Tim and Ken asked me to talk today about aquatic ecosystem health, which is a nice long sort of term that may bemuse some people as to what it really means and why do we do it. So I'll sort of try to cover that sort of both of those subjects this afternoon. And aquatic ecosystem health is a sort of a convoluted term to say biological. What I used to consider biological monitoring when I first started out in my career. In other words, it's monitoring the critters and the life in streams and waterways, rather than filling bottles and doing gaugings and things like that. Really, the question of why do we do it is to bring some sense of reality to sort of monitoring or the resource management we do, which is, are we really having the desired effect of keeping things healthy and keeping diverse and productive communities in our streams? So that was really my short answer. What is it? Monitoring aquatic life, or insects in particular, is how most councils, most people do it. But critical to that is also that we monitor their habitat as well. And if we integrate that there's a diversity of biological life there and there's appropriate habitat for them, then we tend to get a good score. And that's as against water quality, hydrology, or many other sort of diverse parameters, which are really the stresses. And there's been a long focus historically of just measuring those stresses and then hoping that the ecology was okay. And why is it important? Because it measures environmental outcomes rather than these single stresses. Therefore, it demonstrates sort of overall outcomes of our environmental management. And it's really important that we sort of try to do that sort of thing, because we very often get sort of tied up in models or single parameters and encourage yourselves to think that there's one parameter that's actually determining what's making things healthy or not. It's also consistent with the overall purposes and objectives of most of our legislation, sort of policies and plans and things, which right up front normally set some objective statements that say what we're trying to achieve before they then dive into these sort of stressor measures. And that's particularly even RMA with sort of part 2, sections 5, 6, 7, etc., which have all those broad statements of what we're trying to achieve, of intrinsic values of ecosystems and things like that. And probably the most important reason why monitoring ecosystem health is important is it's directly relevant to demonstrating these outcomes to people. As the people of Canterbury out there that you're often interacting with, we try to get them interested in nitrate or all sorts of parameters and things, but they're often things that don't taste. They're tasteless, odourless. You can't see them or anything like that, and they'll go along with that to a certain extent. But at the end of the day, my impression and my career has been looking at people are looking for reassurance that what they can see out there is healthy and that things should be how they should be. So it really comes down to this touch, see, taste, smell, etc. sort of features of people. So what I'm going to cover today, I've just given you the short answer and I'll elaborate on that. What is aquatic ecosystem health? I'll pretty much cover through the New Zealand history of aquatic ecosystem ecological monitoring, which takes us to where we are at the moment, and then I can project a little bit further forward, but also the Canterbury history and the Canterbury drivers of what is biological monitoring through to aquatic ecosystem health, and then where it may go to in the future. I'll touch briefly on what we've used aquatic ecosystem health for, in terms of how we've utilised it, how we've sold it to people, how we're sort of illustrating it to people, and really finishing up with what to next. I'll throw 2 or 3 terms at you first off, and that's biological monitoring is where it all started from, and the word biological is really important, is that we're dealing with biological systems rather than physico-chemical, in other words, physical measures or chemical measures. It then moves on, that's where it started back in the 70s or 80s. It's moved on to the aquatic ecosystem health monitoring, which is the terminologies we put on it as it's become more sort of integrated and more holistic. But there's also another term I'll throw up there on the slide, which is ecological integrity. We're seeing more and more. Some people use it as a synonymous term with aquatic ecosystem health. But there's quite a bit of debate going on in different parties. It was a term more generated in the Department of Conservation sort of domain of more dealing with sort of naturalness and pristineness and nativeness and things like that. So we're struggling a little bit with bringing the two together because it's a very useful term and a useful sort of system within sort of natural conservation estate environments, but I'm having some difficulty bringing it into maybe the intense human environment. So there you go, ecological assessment. I'll put this photo up there with Duncan sitting here, sort of ferreting around in a stream, because Duncan was originally going to be giving this this seminar, but he got tied up on a few different sort of things. So I borrowed a few slides off him. So here's your cameo, Duncan. And the ecological assessment part of aquatic ecosystem health is largely ferreting around in a stream here with a kick net like that, trying to get a broad integrated sample of what's living in the stream. So it's kicking it. What Duncan's doing there is kicking in the stream, disturbing all the insects that are living on the bed of the stream, so they get swept down into the net, and he'll be moving across, backwards and forwards across the stream, both covering a transect plus covering all the different sort of habitats available there. So he's getting a reasonable one-off assessment of what's living in that stream. So it's somewhat qualitative and somewhat quantitative. There's no replication here. We're just trying to put enough effort in there to make sure we've caught some of just about everything that's living there. And these are the critters we're most dealing with, the aquatic invertebrates. You can do this sort of biological monitoring with just about anything that lives in a stream or a waterway. But the invertebrates are the ones that have been chosen and dealt with for the last 20 or 30 years, for a number of different reasons. One, there's a great diversity of them down here, over 600 species probably in New Zealand or something like that, so you've got a great diversity to deal with, and therefore the diversity is one of the important attributes. But also, that great variety of them gives you a load of attributes to deal with. These EPT, which are, to throw some morbid words at you, Ephemeroptera, Plecoptera and Trichoptera. That's a Plecopteran, Ephemeropteran and a Trichopteran, otherwise known to as stoneflies, mayflies and caddisflies, are the high quality insects of running waters. They tend to be sensitive to water quality changes, sensitive to habitat changes, and therefore really good indicators in that sense. They're also the favoured food that a lot of the fish and the native fish, plus also the exotic fish like salmon, like trout or juvenile salmon will eat as well. So what we're often looking for is a diversity of these and a good number of them in the stream, but also a diversity right through. We've got riffle beetles here and the toe biters that some of you or your kids might have come across in streams as probably the largest of the insects you'll see in a stream. And you'll always get some of these ones down the end here, which is the worms, sort of bloodworms or chironomid midges and snails and things like that are pretty much tolerant of just about any degradation in the streams and waterways. And so you'll always get a few of them there. You're just trying to make sure you don't get the community totally dominated by these and lose all of these. And that's the sort of basis of the biological part of monitoring, because these things are diverse, but also the other important point about them is that they're always present into the streams. Although they drift a little bit downstream, they're not migrating anywhere. So if they're not there, they've pretty much been knocked out by the the conditions of the time. So they're very stable and very reproducible to monitor, as against, say, fish and birds that tend to vote with their fins or vote with their wings when the going gets tough. So the other part of aquatic ecosystem health monitoring that's very different to physicochemical monitoring is that you also measure other attributes of the environment, and in this sense it's looking at the habitat. This is just a photo of a stream, and so when you've collected all the invertebrates, you go there and measure a load of different criteria as to how good a quality the stream would be. This one would be largely good because it's got intact riparian vegetation, very little bare cover here, a variety of gravel, cobble and rock types, a diversity of habitats from the sort of swift riffly areas there, a pool down here, a sort of run of smoother water up here, and a whole lot of attributes like that. And if you score those in a concerted way, you can actually come out with a numerical score of how good the habitat is. So this is where we're sort of talking about them, providing a context for any assessment of substrate, riparian cover, macrophytes and algae, what the riffle run pool sequences are, whether there's lots of fine sediment or shouldn't be. Hopefully there won't be lots of fine sediment in the stream, and this all then gives a numerical score of the state of the waterway. So older style biological monitoring was really another type of water quality monitoring, because it largely set out to measure the invertebrates in terms of how sensitive they were to water quality and factoring out flow and habitat effects. And how they got away with factoring out flow and habitat effects was all of that early biological monitoring was done in those quite swift broken water riffles, where the water cascaded down them. The invertebrates were happy in there over a wide range of flows because the water was always swift and aerated. They were also pretty much happy through a wide range of conditions in those riffles, because if there was an inundation of fine sediment and things like that, that wouldn't accumulate in the riffles. And if there was an oxygen problem, say of high biochemical oxygen demand, and that would exert itself worst in pools where you get oxygen levels decreasing away rapidly. But over the riffle, the aeration, the broken water would keep it sort of healthy. So older style biological monitoring that I sort of initially grew up with, and that still a lot of carried around, particularly sets out to factor out all those factors. And so it's largely just another piece of water quality monitoring. However, aquatic ecosystem health integrates all of those stresses to measure an overall outcome. And so therefore we tend to do aquatic ecosystem health monitoring in the smoother runs or over a wider area of the riverbed, where we will see some of those other adverse effects, in other words, sedimentation occurring, algae growing, or aquatic macrophytes getting out of control and starting to smother the environment and things like that. So it's integrating over all of those sort of things. But also then by measuring the habitat assessment that I talked about earlier, you can start to tease those. When you see the invertebrates are not looking healthy, you can tease apart whether you think it's predominantly being driven by water quality, or whether it's habitat, or whether it's other factors. And so it's a much more holistic view of dealing with monitoring. And I'll put this as a sort of stool analogy here, in that biological monitoring would just be basically factoring out those other ones dealing with water quality, but aquatic ecosystem health is like a stool, and it could be 3, 4 or 5-legged, but it's really making sure that it will be responding to the flow regime, will be responding to the habitat quality and the water quality, but also keeping in mind that there may be all sorts of other things like toxicity, predators, exotic species, diseases coming into play. And we tend to want to know those things, because what we're trying to do with an aquatic ecosystem health program is to make sure all of those things can come to bear, so that we know what the overall health of our site, health of our aquatic community is, so we can report that back to ourselves or to communities and say whether we have a problem or not. Another example of this is when we sort of graph some of these attributes together. Down here we've got the habitat score. We can put it on a numerical scale of 0 to 150. And the invertebrate score, which is the biotic indexes, which is what the insects are telling us. And if you plot those two together, if habitat was there, if it was just an interplay between habitat and invertebrates, all the data would fall down along this red dotted line. But if the data tend to fall away into these other areas, you're starting to see you've got other factors other than habitat starting to come. Like if there's really good habitat but really poor invertebrates, you start to say that it's quite likely to be poor water quality or toxicity coming to bear. And so here's some of it. Here's some data from a few years ago on some of those different sites, and you can see it doesn't all neatly sit on that sort of equivalent of that red line. And so in our lowland streams, you can see here we're getting quite a lot of water quality types of problems here in many of them. There's one or two here where we tend to be getting, despite bad habitat, we're getting good invertebrates, which is tending to indicate there's probably good habitat upstream somewhere that's still bringing the good invertebrates in. But what these sort of graphs do is give us a much broader ability to interpret the data and start to try to tease apart what the factors are that's causing maybe not optimal health of our waterways. Well, aquatic ecosystem health as a sort of method has evolved over the years. It originally came from biological monitoring, and some people still operate it as a single metric or a single index. In other words, they measure one attribute of aquatic insects. Often that's the macroinvertebrate community index or one of its variants, which were developed in Taranaki maybe 30 years or so ago. And that macroinvertebrate community index has served New Zealand resource management really, really well. Although it was based on a measurement of what the effect on invertebrates were of organic enrichment. So it was really designed to look at the effect of discharges from, say, sewage treatment plants, dairy oxidation ponds, industries that had sort of organically rich chemical constitutions and things like that. And although it's been since shown to be quite effective at a broader range of other stresses, there are some stresses like fine sediments and things that it doesn't perform very well with. So largely we don't tend to endorse an aquatic ecosystem health type sort of monitoring system based on a single metric. We tend to go on these multi-metric approaches, which tend to measure a whole load of different attributes. In other words, we pick attributes that would show whether the invertebrates are being stressed by fine sediment type sort of stresses. Another one that's whether they're being stressed by organic enrichment types things. Another one if they're being sort of stressed by the wrong type of food and things like that. So, and we try to pull those together, and that gives us a much more integrated approach. And that's largely what we've been doing here in Canterbury for the last few years. Although it's fair to say that there are other aquatic, there are other systems, and they're largely derived models coming from either Europe or Australia with names like RivPACs and things like that, which are largely trying to model what the community should look like in a river of this type with these sort of characteristics. Then when you analyse your sample, you see how far does it depart from that, which is so it becomes a sort of observed over expected ratio. They've been tried in New Zealand a few times, but mostly they don't tend to work very well because I think New Zealand has such a stretched latitude and longitude, and our variety of habitats, say from the east coast to the west coast, and say from Northland to Canterbury, are so diverse that a lot of those don't work very well, and you've got to refine them down into models at smaller and smaller geographic scales in New Zealand. So this is an area where there's a load of different aquatic ecosystem health type methods being promoted by different people, but there's a lot of debate still going on as to what might be the dominant one that may win the favour nationally, especially when we head for national consistency. The other question is, I've been promoting aquatic ecosystem health as a biological sort of monitoring tool, is where does it fit with our other monitoring programs? Where does it fit with our hydrology network, our existing water quality network, where many of our staff go out and sort of fill bottles and send them away to a water quality laboratory. So we end up with sort of three networks, and they've all largely been developed independently. So we have this a network of sites, and the challenge is how to align them, or should we actually be aligning them? And we did it. There was a review carried out 3 or 4 years ago that brought them together, and we've achieved something like 60-65% alignment of hydrology, water quality and ecosystem health programs at the moment. But the challenge has always been, with those are vigorous discussions, is which network should dominate site choice, because they all have an inherent bias. When we develop a hydrological network, we're picking sites that are actually really easy, good and reliable to gauge and don't change very much. When we're picking a water quality site, it's to measure, it's often for ease of access, ease to get to, and it's generally upstream of a bridge type sort of thing that's easy to get to. Whereas Aquatic Ecosystem Health Network, we're actually looking for good average sort of habitat. So you can sort of imagine those three are in tension a little bit. And whereas it's great to bring them all into alignment, there's always this, which one should take precedence? And I think we'll always be debating that to some extent. It's an objective that you'll never fully achieve. So the history of biological monitoring in New Zealand, in 1970, most of it was carried out from the universities, and even in Canterbury you'll see the earlier papers will be Professor Mike Winterbourne and Vida Stout and others that sort of carried out this work benevolently for the people of Canterbury, but it was largely looking at effects of point source discharges. By about the 1980s, some of the councils were starting to get into this, and although it was much more common in other parts of the world, New Zealand was a little bit slow getting into this. In the 1980s, Taranaki started developing methods and doing some of it because of many of the sort of developments that were going on in Taranaki of different industries and things like that. But it really wasn't until the 1990s that biological monitoring really became accepted in most councils as part of the repertoire of monitoring that they do. And to differing extents, it became common, but and around that time the first council started looking beyond the sort of discharge type scenario of what's the effect of discharge of contaminants from the end of a pipe, to looking at the aquatic ecosystem health as how healthy are our rivers. Are they as healthy as we should expect them to be? And so it was probably really after about 2000 that more discussion on aquatic ecosystem health concepts sort of arose. And they were vigorously discussed for a long time, but they've still been very slow to be taken up by most councils, which have still largely stayed with a water quality focus, and biological monitoring being allied to that. By about 2010, there was this increasingly rapid consideration of aquatic ecosystem health or ecosystem integrity, and especially driven by discussions of national consistency as how can we bring this monitoring consistently together, and more drive to say how council's going to operate uniformly so we can report things nationally. And it's probably, I'll put up there at the end, 2015, there's this discussion of the National Monitoring Act, which once again will be a big push for us to be more and more consistent in what we do, as rather than operate independently as separate councils with separate ideas. So in Canterbury, there was probably limited biological monitoring in the 1990s. It was very much plan focused on addressing piped discharges. Most of the when I arrived at the council in the mid-90s, most of the biological monitoring was still very much orientated to, say, the Opihi River Regional Plan, with the plethora of sewage discharges into the river that we were trying to extinguish, or the Waimakariri River Regional Plan was under development at that stage. Very much the same, all meatworks discharges or industrial discharges were the focus. And I'd come from the Waikato where we'd been pushing the boundaries a little bit more rapidly at that point. But it was trying to slowly change the idea and culture a little bit that managing for the ecosystems themselves were an appropriate way to go. And probably it's very topical this year while we've got a drought on, that the 1997 to 1999 drought back then really came to bite us. 1997 to 99 was largely sort of two and a half years of almost continuous drought, where we had dry winters as well as a couple of dry summers, and they really started to bite, and there was some very vigorous discussion going on about fish salvage, fish kills, unhealthy rivers, etc. And that really polarised a lot of thinking. It came to a head in several board meetings between the council at the time and a lot of stakeholders about how healthy are our rivers after all of this drought period, and things like that. And our somewhat embarrassed responses, we really don't know, because we really don't do any biological monitoring. We know there's fish salvage going on by Fish and Game and things like that. But and that really triggered a 100k budget that came out of the woodwork at about that time to address that issue. And that took up a lot of my time for a number of years at that point, was to say, how can we put this sort of work in place when that drought gave us this opportunity. Maybe I'm not saying that this drought at the moment is an opportunity in other sort of ways, but you know, out of those sort of stresses often do come opportunities. $100,000 wasn't a terrific amount of money to put in a biological monitoring program for a region the size of Canterbury, because we are pretty much the biggest regional council in New Zealand. And so there had to be a bit of innovation. We were lucky with the University of Canterbury being on our doorstep, with the likes of Professor Mike Winterbourne and others being very well-regarded aquatic ecologists there. And we tended to work in with those to put in a program that was utilising University of Canterbury students and some supervision from the University of Canterbury staff resources. So this allowed us to streamline biological and habitat methods by hiring students that were already partly trained by the university over the summer, standardising a method, and that was worked very well over the last 16 years. We still hire students, most of them from University of Canterbury, and many of them have gone on, and I'm sure there's several of you in the audience as well that have been through that pathway. And an intriguing part of that is that's sustained with that summer program of students as well. It's got multiple benefits. One is it's very cost effective and cheap. Two, it is training up a number of people in these sort of methods that then find themselves into all sorts of different jobs. And it's sustained itself very, 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 very well. It's intriguing that very recently, in the last couple of years, a whole consortium of North Island councils have realised they have to beef up this sort of monitoring themselves terrifically. And they've pared it down as much as possible, streamlined it as much as possible, and managed to coordinate recently that they can get their samples now processed for around $200 a sample, and they've all gone in together. The intriguing thing about that is what we streamlined back in about 15 years ago and is sustained now is actually costing us via the summer students process and doing it all ourselves. It's still costing us well under $100 a sample. So those other councils have now largely come to our methods by a different pathway, and yeah, it's really gratifying to see what we developed there and how cost effective it's been. So what do we do? I've done a lot of talking today. Here's our aquatic ecosystem and health sites by plan river type or rec river type. And so we put as many sites as we can in so we can get a snapshot of. And we've got a cluster of Banks Peninsula type rivers here. All the bright red ones are our lowland streams, which we've built up over the years. It's where a lot of the rubber really hits the road and where the pressures come to bear, including a few alpine rivers, the green ones up the top. So we're taking a snapshot of maybe at least 5 different river types, so we can see which ones should be healthy and are they staying healthy, which ones are under a lot of stress and are we stopping them getting any more stressed, or are we managing to keep them back. Also in recent years we've rationalised those to make sure that they're adequately represented in the different CWMS zone boundaries and things like that, so there's a reasonable representation now so that they service all the different zone committees. I've already shown you that graph before. I'm not sure why it's still sitting in here at this point, but it's one of the ways we analyse the data. What our staff have been doing in recent years is every year is now pull the data together to service different customers. And now the zone committees are one of the important ones. So here's Selwyn Tewahora Zone Committee. Every year we'll pull the data together for each of the zone committees and present it in this manner, where each of these symbols is illustrating the invertebrate grade, in other words how healthy the ecology is, on the left-hand side of the figure, and the habitat grade, which is how healthy the habitat is on the other. And this is a sort of first cut illustration of how things are looking, and this may change slightly each year. In terms of longer term trend, we've been producing these sort of figures for a long time. This is the Selwyn Te Waihora spring-fed streams. So when we've got something like 10 or a dozen streams, we'll split them up into what proportion of them are in very poor condition compared to what proportion of them are in very good condition. I'm always hesitant of these graphs that have been used by different chief executives in the past or all sorts of other stakeholders, as they're quite illustrative, they tell a story. I'm hesitant about them being called a trend. Well, they're largely showing a pattern of where things are moving, but we get quite a variation year by year that can be driven by climate, can be driven by all sorts of other things. Once again, you seem to hear that there seems to be a growing amount of red, so therefore there's more stress coming on the lowland streams. There's more of them in the very poor condition. Probably the up along the top, this was a particularly bad year, but things bounced back a little bit, so you can interpret these in all sorts of ways. But there's a small core of very good quality streams that largely stay in that sort of state. But these are just the broad summary type results, but we can tease them apart in all sorts of other ways. So that's given you a sort of teaser of what our ecosystem health program has been, where it's come from, where it's at the moment. So the obvious question now is, where do we go to from here? And all the indications is that the program's still supporting us very well, giving us very useful data, that we're able to communicate with many of the different stakeholders, the public, our own organisation and things, and again get some reasonable indications of where the stresses are coming from. So I'd largely say the first thing where to from here is keep doing what we've been doing to consolidate what's now 16 years of data. We're in this luxury now of Environment Canterbury having 16 years of this sort of data and seeing where we've come from and seeing where we go to. We're in a much better position than most other councils that haven't had a program like this, and are still having some very awkward discussions about what things used to be like and things like that. It's also important that we now continue to interact with and influence national method developments and national reporting, because we've got a big investment in what we've been doing. It's been really interesting to see how a consortium of North Island councils that were always critical of what we've been doing have recently got together and tried to streamline ecosystem health monitoring, come up with a system that looks very, very similar to what we've been doing for the last 16 years. So that's gratifying, but rather than resting on our laurels, it's working with them to make sure that they're learning from what we've learnt, and we're not being pushed too vigorously for all the wrong reasons to change our program and start to corrupt this 16-year data set. But also it's looking for opportunity to expand the approach into other water media or other values. So, given the success, it's quite important to look with other water media. The Canterbury Water Management Strategy is a fundamental tool we've got now. It's a partnership with communities and stakeholders and things like that, and the district councils. So therefore, but it's more than just small wadable rivers and streams. So other water media, wetlands particularly, we've supported in the past few years some NIWA work on biological monitoring of wetlands and how that could sort of work, and also working with other groups in the council here, like Philip Grove's wetland work as well, to sort of say, can we work up through that, and there are remaining wetlands in Canterbury, how can we monitor them better to have a better indication of where they're going. Lakes, we've established a water quality monitoring on lakes really well in the last few years, but really that's got the same issue. We've worked in with biosecurity and NIWA to have a lake spy, which is a submerged plant index, which gives us a broader indication of how healthy lakes are and what might be causing stress on them. But we're just entering now into some assistance with NIWA that's starting to measure the zooplankton, which is the sort of water fleas and the copepods and all the insect life that sort of filter out the water in lakes. And that's got parallels to everything I've been talking to today, that working with that is a great way of seeing what's actually causing lakes to change. You know, it may well be that nutrients are making lakes greener, but you can have a pushback the other way, is that we've recently been getting exotic daphnia and exotic introductions into our lakes that are very efficient at filtering out algae and things like that. So it's important to know if those drivers are happening as well. Hapua, which are the gravel bar blocked mouths of many of our Canterbury rivers, are mentioned quite broadly in the Canterbury Water Management Strategy. We're struggling a little bit to figure out how to monitor them, but they're of particular importance to iwi. They're of particular importance to communities that have large batch communities of those mouths. So that's another whole area really begging us to look more detailed what we should be monitoring down there and how we should be managing those environments. And I hesitantly put groundwater there as well, not as a groundwater scientist myself, but I know some of you groundwater people will have come across Graham Fennick and others that very emotively like to describe all the groundwater fauna that live in groundwater. Groundwater isn't a sterile environment, it is quite a dynamic environment. It does have an ecosystem down there, and whether there's a place for any of that monitoring in the future. The other one is other animal groups, and particularly as with our partnership with iwi and things like that as well, it's deciding whether we should be moving on into monitoring megafauna, particularly potentially important mahinga-kai species like crayfish or mussels that we won't pick up in our aquatic ecosystem health programs, but are probably really important to monitor as well. Fish, which are another challenge themselves, that's where I came from. Many in our team would love to be spending much more time monitoring fish, but they're a challenge in themselves because they're migratory. Many of them have to go out to sea at different times, and they're more difficult to monitor, but that's not a reason not to. And bird populations that many of you will have interacted with, say the likes of Francis and the riverbed nesting birds issues. I'll give you another cameo, Duncan, of charismatic megafauna. That's Duncan catching a charismatic megafauna, isn't it? Which I've just said, we'd all love to be monitoring fish and things like that, but they are highly mobile, they're migratory, they're hard to catch, and reproducibly catch. There's a lot of regional endemics, so therefore you'll get lots in one place and nearby none, but that's natural. And they're very resource intensive to monitor, a lot more resource intensive than, say, the macroinvertebrates I've been talking about today. And so often it's more effective to initially monitor the stresses, which as a council we've been doing a lot more of in recent years. That is a fish passage program with our biodiversity section, fish screening and trying to beef up that sort of protection, and managing critical habitats like whitebait spawning habitats or critical salmon spawning habitats and things like that. Once we've got those ticked off, then a fish monitoring program is certainly a possibility, but I think that's probably the best way to go first. And so, I'm not sure about the time, but I've probably been talking way too long, but in summing up, our ecosystem health type programs have a critical place in our monitoring programs. It's where I've spent most of my career and most of my life working with them. I'm a firm believer in them because they are the final outcome of what we're all trying to achieve by smart and sustainable resource management. They give context to whether we're really achieving those big outcomes, and they're specified up front, as I say, in RMA, in Table 1 in the front of the Land and Water Regional Plan, which are very much, these are the outcomes we're hoping to achieve, and even in the National Policy Statement for Freshwater Management that the government's produced recently, the NOF tables are very much, their starting point was ecological outcomes focused. And so really they're essential for helping us decide whether we're on the right track, whether we're getting to the right places, but also can help us explain what the big issues are we're really trying to tackle. Is it habitat? Is it water quality? How is it interplays of those sort of things? And finally, a really important part that I keep coming back to is it's a program, and it's putting things in a context that are meaningful and understandable by people, because at the end of the day, when people are coming complaining or wanting to have dialogue, whether it be with us as staff or with zone committees and things like that, they're really asking, are these environments healthy? Are the right things living in them? Is it a balanced system? And I think these aquatic ecosystem health programs are therefore a really important foundation for being able to do that. And we'll finish at that point.
Anybody have any questions? I'm sure that.
On your last statement, thank you for the interesting information. Good. Given your last statement, I'm interested in how useful citizen scientists would be in supporting the work that you do. I know there's a lot of community groups or schools that do some form of monitoring. I'm just wondering whether there's any kind of engagement.
Yes, I thought there's key examples like the Sticks Living Laboratory and things like that, where I think they do a lot of this work, and I think there's greater scope for communities and citizen groups monitoring biological systems than there is actually filling bottles and sending them off to a laboratory or getting numbers, because it's really hard to get them keyed into that and really believing in it type sort of thing. I think yes, I think there's a really big place for community groups, community monitoring and things. Certainly there's quite a large NEWA program at the moment trying to coordinate professional ecosystem health type science with community type ecosystem science and seeing how reproducible and whether they produce the same things and are just as reliable. And I think with the right training, hopefully they will be.
Yeah, that's given them the right training. And
Yeah.
But a supplementary question, how does this information feed into LAWA, the online community?
That's another whole question to some extent. Is LAWA, when it first got operating, the first module was physico-chemical water quality, and that's up and running and getting really good feedback. It would be really great, and I think it's working towards getting some biological data up. It'll probably be just a single parameter to begin with, like MCI or QMCI, and getting that up, because that's the sort of thing that both can work on. But it's always a little bit of a challenge getting into that. It's the same as the National Objective Framework from government as well. We were really hoping that biological measures would get into that, and I'm sure they will, but there's a little bit of fear and hesitancy about that getting up there to begin with, because it's an outcome measure rather than a stressor, and therefore there's not so much certainty what the result will be, but it's very relevant to people and very important for people.
Yeah, I just had a question in relation to the grading. I wondered how the natural state of the different rivers works with that grading system, and whether in some places the natural state, or whatever that is, in a place would be very poor, and how that you can kind of communicate, if you can't communicate trends, how you can communicate patterns in relation to the natural state of the river monitoring site.
No, that's a very good question. That's a very big, important context question as to what delineates poor, fair, good, etc. With our multi-metric systems, we've sort of borrowed very heavily from the American system, and using a core of reference sites for each of those water body types. And for particular ones like lowland streams in Canterbury, where there's no pristine lowland stream that you'll find anywhere, there's been a lot of soul searching as to what a reference condition should be, and in that sense, the philosophy through these programs is very much what could it reasonably be, given the acceptable extent of development and things like that. So we're certainly very good. Is not representing what would be in a national park or pre-European or anything like that. And so there has been quite a bit of proofing of concept there as well, is that the reference condition is what's reasonably achievable, and partly what's still present in the one or two really good environments. It's very easy in a mountain stream where you've got genuine pristine ones, in say lowland streams or even intermontane basins and things like it. It's very difficult to find out what would be a baseline. So we're being quite realistic about that. Yeah. Sorry.
I mean, these indices are good for informed policymakers, but I haven't seen a lot of them come through in resource consents. Do you think there's a place for them in consents, or are they too sort of difficult to actually put a hard and fast number? And it might be exactly the same reason why you're not seeing them in the NOFs, or you didn't see councils adopt them.
Another good question, and one that's vexed me at times over the years, is that I remember back in Waikato, planners were trying to phrase conditions of the MCI shall not decrease by more than 10% or something like that. These indices are integrating communities over a reasonable period of time, so they're giving us a direction of change. They're quite difficult to actually put in place in terms of hard and fast consent type sort of targets or compliance type sort of limits. So I'm not aware of many of them that have gone them.
Just too hard.
I think there's too much uncertainty. They're giving a good direction of change and good direction, so therefore they're good sort of higher level policy type instruments, which is why we've often used them with terms like outcomes and objectives and things like that, rather than sort of more explicit targets. And I think that's fair to say why government has been quite hesitant about throwing them straight away into the NOF and things like that. Although
But if you
I think they do that. There is a potential place, but there's some bravery needed with that.
and maybe for a longer term, 35-year consent, you could run something in that for a shorter term.
Adrian, and I want to assure you this is not a personal question. Although I might just make a comment to reinforce something at the start. I can remember when we set this program up 16 years ago, with a fair bit of nervousness on the part of some decision makers about handing over such a generous sum to the body. But we've now done it for 16 years. It's a $1.6 million investment, and that is actually for the data we put out of this program. A pretty cheap kind of, it's a pretty good investment. You can say what we spend on some other monitoring programs. But I use the kind of data advisedly because the question I've got is that in that data, and a good example would be that Selwyn White or a Springfed stream spread. Well, there's obviously a huge amount of information. There's a story in there. Yep. But my question is, how good are we at telling that story? And what do we need to do to get better at it? Which implies that I don't think it's doing very well.
Yeah, very. I think there's a vast amount more information to mine out of this. We've, and it's probably fair to say that many of our monitoring programs, not just ecosystem health and things, but even chemical and things like that, could be mined a lot more to illustrate more to us. It's how much we're being asked for and how much resources we've got to actually dig it out. I don't know. We keep, as we bring people like Duncan on board and Michael on board and things like that, we hope the enthusiasm of youth as they'll dig in and mine these data and things like that as well.
Students,
But yeah, and I totally agree with you. There's lots more data, lots more information there, and it can be churned up in all sorts of ways.
Just to clarify, I was also thinking, particularly in relation to that slide, you know, what's the underlying message in that slide? And how do we get the zone committee gets very interested in that? And how do we convey that?
Well, we've been evolving that at times, like that most recent slide of which has got the symbols, which is including habitat and biotic index together, is almost begging to have questions asked as why for some of those sites is the habitat good but the invertebrates poor, or vice versa, and it's leading to questions like that. It's maybe also saying that those patterns aren't consistent across that zone, so therefore there's different pressures in different water bodies. In other zone areas, there are very consistent sort of messages coming through. I don't know. Am I getting to where you?
Yeah, I think so. I mean, I just think the message is that there's lots of data.
It's partly for us to try to keep selling it more, to get the questions, to get dialogue.
And
it's having a conversation.
The dialogue, you're right, it's actually asking the zone committees how they want their information. What gives meanings? What should we get on the website? What's useful for our colleagues at Aquatic use this information and policy development and enforcement, whatever federal court of action endeavour they
I was really following on from what Kim was saying about the story. It's for me thinking about the CWS and zone committees that have been very much focused on initial ZIPs and then into zip addendums and planning mechanisms, but sooner rather than later they're going to come out in the end of that and they're going to start wanting to see data that's information that's telling them are they reaching the outcomes and goals that they've set in their zips. And I guess the question I have is that information, is your information in a state and in a shape that's going to be able to tell that story to them? Or are there things that we might have to adapt or improve?
We're certainly packaging it, like some of those figures in the presentation, we're trying to package it in zone packages type sort of thing and making it available. It's trying to get into that dialogue with the zone committees is part of the challenge, but we certainly are packaging it to them because we'd like to have dialogue with them, we'd like them to be utilising it, asking questions, having discussions about it. So it leads them in certain directions, or they start to lead us into more monitoring they like or more questions they like answered.
I'm going to stand up because I should have been in the front. And I'll wrap it up, I suppose. And I apologise, I was meant to be here to introduce Adrian and start the seminar, but I was rushing to get back from South Canterbury and then got upset by the Foo Fighters being in town, which I didn't realise, funnily enough. I was just partly to what Davo was asking there, I was just thinking about, of course, one of the CWMS targets says, and I can't remember the figure, but it's a certain percentage of lowland streams that ecosystem health is above good or very good, or I just can't remember the target, but there is actually in the CWMS a target directly linked into this ecosystem health monitoring program. So that it sort of gets a bit towards what you're saying, and also partly to what you were saying, Richard. I think
the first target was one was going to be improved, and then a period later a percentage of them will be improved and things like that. Right. So it's staged. Yeah. So it's certainly, very much so.
Just before we wrap up, I was just going to ask you, Adrian, is there a broad picture you can tell though? You showed that one of the lowland streams and you said, we're a bit nervous about showing some of this. What's a broad picture you can tell about Canterbury from our ecosystem health monitoring? Perhaps different river types and things like that. Is there something?
Well, on the other extreme, the alpine rivers are pretty much looking after themselves. On one extreme is, apart from probably the failure of nor'westers a little bit this year, we're probably seeing some of our higher alpine rivers probably in their poorest health, but probably for climatic reasons this year. But largely they look after themselves, and they're sitting out on one extreme of, and I could have bombarded you with lots of data showing that pretty much alpine streams are almost all in the good and very good category. The lowland streams are much more depressingly in the poor and very poor, but we're holding on to a few good ones, and it's probably a parallel with the American system, where I think the question on reference conditions and what makes a good lowland stream, in the American situation, most of the reference conditions for lowland streams are ones that have had community groups and intense recovery programs going on suddenly become the best reference conditions in the region. And I think we're probably hoping that in some of our areas, that's where we're going to really making the progress with community groups trying to improve them. In the middle ground between them, hill-fed rivers. I know we used to particularly present lowland streams and hill-fed rivers as the two categories that were in the annual plan sort of targets, because they contrasted a little bit. Hill-fed streams, by and large, are in reasonably good condition, but are increasingly suffering from probably flow recession as much as anything. We've still got a lot of very good waterways, particularly those that are draining from further west, further in the inland areas.
No, that's good. I think, unless anybody's got one last burning question, we probably should wrap it up, in which case we will. And I just want to acknowledge Adrian for this for several reasons. When we were designing this science seminar series and we were sort of working through the different ones and it got to surface water science section, I was really keen to have something on the ecosystem health monitoring program because I think it is the most important monitoring that we do in the council, and I'm not trying to talk down anything else that other people do, but I do seriously think it is probably our most important monitoring program for reasons that Adrian's outlined in terms of the visibility of it and that people can understand it. So I think it's tremendously important. Adrian was instrumental in getting it going, so it's great to have him to have seen that through. And I just think the other wonderful thing about this program is that in the surface water science section, there are 5 people who are in the science group now, 2 of whom I can see here today, and I've probably missed some others, who came through this program, who were students and worked on this program, and then have come through as scientists in the group. So it's been a tremendous training ground as well for us, and has really helped with our links into the university. So in so many ways, it's just a fabulous monitoring program. I just want to say thank you, Adrian, for the seminar, and just to remind you that we will be having another one in
25th of March.
25th of March. We've got the topic right completely sorted out yet, but we'll be letting people know. So the 25th of March, and thank you very much, Adrian.